Hi, this lecture is about cell division, namely mitosis and meiosis. Those are two different types of cell division that have two different goals. Some of the processes are similar and some of the terms used in these two types of cell division are the same. I'll warn you, there is a lot of terminology associated with this unit. On your study guide, you have a list of terms. I don't expect you to be able to write out a definition for each of those terms. Just know what that term is referring to, and you should be able to use that term in the story of cell division. Again, I would never ask you to write out a definition for those terms, but it's important to understand what they are and how they relate to the story of cell division. Two different types of cell division. Mitosis is what we call division of the somatic cells. This term somatic cells, soma means body. These are the body cells. Every cell of the body except for eggs and sperm. Eggs and sperm are called the gametes. Meiosis, on the other hand, is division of a cell to produce gametes. Gametes are eggs and sperm. All sexually reproductive organisms have gametes. Sometimes they're in the same organism. Think of an angiosperm, a flowering plant. In that flower are reproductive structures that produce eggs and produce sperm. Yes, plants have eggs and sperm that come together for fertilization, but they happen to be in the same individual. Different individuals can cross-pollinate and fertilize each other, but you could also self-fertilize. In animals, most animals are some exceptions, such as certain parasitic worms, but a lot of animals have gametes in two separate organisms. Again, not all animals have that, but a lot of animals do have individuals that produce eggs and individuals that produce sperm. In mitosis, the goal is to produce genetically identical cells from the original cell, whereas in meiosis, the goal is different. In mitosis, that organism starts with a cell that has what we call the diploid number of chromosomes, the double number. What do I mean by diploid number? Just to kind of step back in your memory for a second, all sexually reproductive organisms start from an egg and a sperm, and fertilization occurs. And that produces that very first cell called a zygote. That zygote came from two other cells. So it now has double the number of chromosomes. In other words, this egg had the N number of chromosomes and the sperm had the N number of chromosomes. And now this zygote has the 2N number. I'll use humans as my example here. In humans, the egg has 23 chromosomes and the sperm has 23. And when those come together, they produce this cell that has 46. So we would say in humans, the diploid number of chromosomes, or the 2N number, is 46. Half of that is called the haploid number, which is the same as the N number. Half of that is 23. In mitosis, you're starting with this cell or a cell that came from this cell. And those all have 46 if you're human. If you're another organism, it has the double number, the diploid number. Mitosis starts with a diploid cell that has the full number of chromosomes. One cell division produces two genetically identical, sorry, that says identical, squeezed in there, two genetically identical 2N cells. Both parts of this statement are really important. Chromosome number stayed the same, started with 2N and ended with 2N. So if this was a human cell, that would mean start with 46 and end up with two cells that have 46. Genetically identical. All cells of the body need to be communicating with each other and producing proteins the exact same way from the same DNA recipe. You wouldn't want your skin cells making proteins 20 different ways with 20 different DNA recipes. They all need to be making proteins the same. The DNA needs to be identical in all of those cells. And there are checkpoints to make sure that happens. So chromosome number stays the same, and those cells are genetically identical. In meiosis, the goal is going to be different. In meiosis, the organism starts with a 2N cell because that's all they have. So from that zygote, everything is 2N. Every cell of the body came from that original cell, including the precursors for eggs and sperm. 2 rounds of division. And those are called Meiosis One and Meiosis Two. And I'll just put M1 and M2 for short. And 2 rounds of division are going to produce 4 cells that are all haploid. And they are all genetically different, not genetically identical. 4 haploid cells, and they are all genetically different from the original cell and from each other. We are going to talk about how that genetic difference in the gametes is achieved and why that's so critically important to survival of a population of organisms, that genetic diversity that comes from meiosis. Meiosis produces gametes, whereas mitosis produces somatic cells. 2 different types of cell division. The processes are similar and a lot of the terms are similar. But because meiosis is going to be a reduction division, the process is going to be a little bit different. Cutting the number of chromosomes in half. Why do we need to cut the number of chromosomes in half? Well, any sexually reproductive organism, what if they didn't? Fertilization occurs and now there's an individual with a 4N number. And what if they mate with somebody that has an 8N number? Now you're at 12N. Every single human on Earth would have a completely different number of chromosomes depending on how many times your ancestors had reproduced. So this isn't what happens in any sexually reproductive organism. It's very important that each of these gametes have the N number of chromosomes. And I just realized this 4N, this looks like that's a 4N cell. This should be 4 times N. In other words, 4 haploid cells. A couple of terms that are really important before we get started. And I will show you at the end, I'm going to show you some slides. I'll be honest, I've recorded this whole thing where I was writing on slides and the slides just, you couldn't read it as well. So I'm going to write on my notes here. And then at the very end, I'm going to show you some slides that will hopefully, just be a review and be a better picture of what I'm showing here. Let's assume this is chromosome number 1 of some organism. It doesn't have to be a human. And I'll typically draw the cartoon version, something like this. This entire thing is a chromosome. And this one piece of DNA on the chromosome is called a chromatid. And this one is also called a chromatid. When they're attached like that, it's because they are genetically identical. Those are two genetically identical copies of each other. One of those copies was made at some point in the cell cycle that we're going to talk about today, called the S phase. When they're attached, they're called sister chromatids. And they are genetically identical copies of that DNA. Sometimes in the cell cycle, a chromosome has two chromatids. So we would say this chromosome number 1 is made of two chromatids, and they are sister chromatids. They are genetically identical to each other. Sometimes chromosome number 1 is going to look like this. So this could also be chromosome number 1, but it just has one chromatid. Depending on where this cell is in its cell life, sometimes it will just have one of each, one chromatid for each of the 46 chromosomes. And sometimes it will have two that are attached, depending on where in the cell cycle that cell is. And you'll see that when we tell the story today. So if this was a human cell, you would have this times 46. And sometimes in the cell cycle, you would have this times 46. So you'll see me referring to that. When a chromosome is drawn, you usually have this little constricted area here where we show the two chromosomes attached to each other. And that is called the centromere region of the chromosome. So this region right here, where it's a little constricted and we show them attached, that's the centromere region of the chromosome. In this region are two really important structures, these little ring structures, to which microtubules are going to attach to pull those two sister chromatids into two separate cells. The whole reason this cell made a genetically identical copy of its DNA is to pull that into two separate cells during cell division and make two new genetically identical cells. In order to pull those apart, microtubules are going to attach to these ring structures. Those ring structures are called kinetochores. So it's called a kinetochore, but it's spelled kinetochore. So this is pronounced ka, and that's an R. Sorry, it's a little messy. This ring structure is a kinetochore, and this ring structure is a kinetochore. Microtubules attach to the kinetochores during cell division. Remember that microtubules were part of the cytoskeleton when we talked about cell structure. And these microtubules are going to be a really important part of the story because the microtubules are what are going to pull those two sister chromatids apart during mitosis. And they're going to play a role in meiosis as well. And those microtubules attach to this ring structure. It's in the centromere region of the cell, but it's this ring structure called a kinetochore. Again, I'm just trying to get a lot of vocabulary out there right now. Those microtubules are produced in a structure called the centriole. The centrioles, sorry, my goodness. The centrioles are called the microtubule organizing center of the cell. This is where the microtubules assemble and start emerging and eventually attach to the chromatids. I'm going to draw a picture of that for you right now. Let's assume this is a cell. And it's getting ready to divide. And so the nuclear envelope has broken down and it's starting to disappear. And you'll see that happen when we talk about cell division. And here's a chromosome. And it's copied its DNA and attached that copy. So we have two sister chromatids. And the goal is to pull those into two separate cells. Here's another chromosome. It's a little bit smaller. It's a different chromosome number. And in order to pull those apart, microtubules are going to start forming. I'm just going to draw these two circle structures here and call this a centriole. There have to be two, because from these centrioles, these microtubules are going to emerge, and those microtubules are going to attach to the kinetochores on each of these chromosomes, and they're going to attach in such a way that one sister chromatid is attached to microtubules from one end of the cell, and the other is attached to microtubules from the other end of the cell. And eventually, those two are going to get pulled apart and moved to two opposite ends of the cell. Same for this one. One of the sister chromatids is going to be attached to one end of the cell, and one is attached to the other end of the cell. Kinetochore, kinetochore, microtubules are the black things. Those microtubules come from the centriole. When you have a lot of these microtubules coming out of the centriole, this whole complex here is called a spindle. So during mitosis, you'll sometimes see this called the mitotic spindle. It's all the microtubules together, and they look like a spindle on a weaving loom. You know, these are some old terms that came from back when people did things like weave their own cloth. Cloth is still woven, I guess, in some areas with a spindle, but that's where that came from. A lot of terms there. Also, it's important to realize a couple of things about our DNA. When you see individual chromosomes like this, that is really highly condensed DNA, but the DNA is only like that during cell division. So you're going to see in the cell cycle that there are certain times where that DNA is tightly condensed in these little nice, neat packages, but during most of the cell cycle, it's not like that. So let me show you two examples of that, of what I'm talking about. These are the last two terms I'm going to give you until I start telling the story. And then, of course, I'll have more terms. Two different times in the cell cycle. In this first example, the DNA is all stretched out, uncondensed, looks like spaghetti. You can't make out individual chromosomes if you look under the microscope. And at that point, that DNA is called chromatin. Uncondensed DNA. And this is how the DNA exists during most of the cell cycle. The reason for that is we're constantly making proteins. The number one job of that DNA is it's the recipe for how to make every protein. In order to be able to get in there and make that little piece of messenger RNA to go out and have a ribosome assemble and start translating that into a protein, that DNA needs to be all stretched out and uncondensed so an enzyme can get in and read that DNA recipe. For this cell to be doing its cell job, It needs to constantly be making proteins. And so most of the time when you look under the microscope at a cell, it's going to be uncondensed DNA called chromatin. At some point during the cell cycle, if we need to start pulling sister chromatids into two separate cells, and it's all this tangled mess like that, that's not going to be possible. I mean, under my desk right now, I have this bin that is just a bunch of cords in there all tangled up together. When I need to pull one out, it's almost impossible. I mean, think about before AirPods existed and you had to use your headphones for your iPhone and they're just like all tangled up. Imagine like 10 of those together, or imagine 46 of those together. It's almost impossible to separate them. So the DNA starts condensing, it starts wrapping tightly around these little protein beads called histones. So the DNA condenses before cell division. It wraps around protein beads called histones. So imagine you have this stretched out DNA and it's going to start wrapping around these little beads, really tightly wrapping. And then it's going to wrap on itself again and again and again. And eventually you're going to go from this stretched out DNA to this really tightly coiled DNA that looks like this. And interspersed in here are all these little protein beads that it wrapped around. And if that's attached to another one, it would end up looking like this. So individually, when they're all stretched out, it would look like this. And then when it condenses, it's going to look like this. And it's much easier to pull this into two separate cells than to pull this all tangled up with its friends into two separate cells. So when the DNA is uncondensed as chromatin, it looks like this, but then at some point in the cell cycle, it's going to condense and it's going to look more like this. And then you'll see individual chromosomes. So typically we call these chromosomes, even though the chromosomes are here, but they're uncondensed. When they're condensed like this and tightly wound, we typically call those chromosomes. Which is kind of silly because these are really individual chromosomes also. They're just uncondensed DNA, chromatin. Okay, now it's time to start telling the story of mitosis and the cell cycle. And I'll be referring back to the cell cycle a little bit when we talk about meiosis, but we're going to start by talking about mitosis, division of somatic cells. There's this imaginary clock that makes up what we call the cell cycle. The cell cycle is incredibly important to understand. When the cell cycle goes wrong, you get cancer. And sadly, every single one of us, if we haven't been already, will at some point in our lives be touched by cancer because either someone we know and care about or ourselves will get cancer. Personally, my mom died of cancer when she was 50, and my youngest sister was 11. I'm the oldest of 6, and it had devastating consequences for our family. Since then, I've lost grandparents to cancer, friends to cancer. My best friend's husband died at age 40 from melanoma. Cancer is a horrible, horrible thing. And a lot of you are going to be healthcare providers, and it's important to understand a little bit about what goes wrong. And to understand how things go wrong, you need to understand what's supposed to happen. What is supposed to happen when things are working right in the cell cycle? The cell cycle is the life of a cell from the time it divides from a previous cell till it itself divides again. And we typically make it look like a pie chart. And the length of each of these stages really depends on what type of cell we're talking about and whether that cell's functioning normally or if it's a cancer cell, because the cell cycle for a cancer cell is going to look a lot different than the cell cycle of a healthy cell. G1, S, G2, and M are the 4 major stages here. Collectively, G1, S, and G2 make up what we typically refer to as interphase. And every cell, if it's a healthy normal cell, spends most of its life at interphase. During interphase, the cell is mostly doing its cell job. So if this is a cell of the pancreas that makes insulin, it's mostly making insulin during interphase, doing its cell job. It's making a lot of proteins, carrying out basic cell metabolism, all of the things that that cell is supposed to be doing. So that means that DNA is uncondensed. And it appears as chromatin under the microscope. So that cell can make proteins. At the S phase, it's going to copy its DNA to make those sister chromatids. To do its cell job, that DNA needs to be uncondensed. G1 starts when a cell has gone through mitosis and divides into 2 new small genetically identical cells. And each one of those cells is now going to start its life by going into G1 and then S and then G2. And eventually each one of those cells is going to divide itself again. So they're going to go all the way around the clock, and eventually each one of those will divide again. So when this cell cycle clock starts, each of those new little baby cells is starting G1. So the first thing that needs to happen, obviously, is some cell growth in G1, which is going to include some organelle replication. So whatever this cell's job is, it's going to make a lot of those organelles. If it makes a lot of proteins, it's going to make a lot of ribosomes. If it's making proteins for export, it's going to need a lot of endoplasmic reticulum for those ribosomes to attach to package those proteins for export, a lot of Golgi, etc. But it's mostly just doing its cell job, just like it is in all of interphase. So it's making proteins. Now, at some point, it's going to be time for this cell to copy its DNA, because right now, that cell just divided from a previous cell. So this cell going into cell division, every one of its chromosomes looked like this. And now that DNA got pulled into 2 separate cells. So each of these cells got 1 sister chromatid from each of those 46 chromosomes. And so now in G1, every single chromosome is just 1 chromatid. So we have that times 46 if we're talking about a human cell. 1 chromatid for each chromosome. For this cell to eventually divide again and make 2 new cells, it needs to double the amount of DNA. So it's going to make a copy of each one of those chromatids. And that happens in the S phase of the cell cycle. This is the phase of DNA replication. And at the end of the S phase, every single chromosome is going to have a copy attached. So we'll have that times 46. Later in the semester, you will learn exactly how DNA replication happens during the S phase of the cell cycle. All you need to know for now is every single chromatid is copied. So we now have 2 chromatids. So every chromosome will have 2 identical copies of DNA attached. Those are called sister chromosomes. So as we go into G2, every single chromosome looks like this, times 46, if this is a human. And then in mitosis, those are going to pull apart into 2 separate cells, and those 2 cells will start their new cell life again. Before that DNA is copied, it's very important for this cell to check and make sure all that DNA is correct and there aren't any mutations or any major damage to that cell, because you don't want a bad cell making copies of itself, and now you have 2 bad cells, and then you get 4 and then 8 and then 16, and before you know it, you have millions of bad cells with the wrong DNA recipe. Before that DNA is copied, there is an important checkpoint, and that is called the G1S checkpoint. Sorry, my diagram is getting really constricted here. That G1S checkpoint, sometimes just called the G1 checkpoint, is really checking a couple of things, but the main thing it's checking for is DNA mutations. Are there any DNA mutations? You don't want bad DNA making a copy of itself. So if we had a chromosome that had some damage to it, you don't want to make 2 copies of that. While that checkpoint is happening, the cell cycle halts. In other words, going around that time clock, time stands still. That cell is frozen in time in terms of it's not progressing to the next stage of the cell cycle. And when that cell cycle halts, that's called the G0 phase or the G0 stage. Those 2 terms are kind of used synonymously. Cells do not progress toward cell division. There are actually a couple of reasons that a cell might be in G0. During development, certain cells go into G0 permanently. As you probably have heard, your brain and spinal column, your brain and spinal cord, once those are fully developed, if damage occurs, those cannot be repaired. It's because those cells are permanently in G0. Figuring out how to get those cells out of G0 is a big part of research to learn how we can repair damage, to get those cells in the brain and spinal cord to go back to a point where they could replace themselves and repair. So some cells permanently go into G0 and cannot divide again. So this would be brain and spinal cord. Some cells are in G0 until they're damaged. And your muscle cells are a good example of this. If you're just sitting around all day, your muscle cells aren't getting bigger, right? They're not dividing and making more cells. But when you work out, when you do a hard weight workout, for example, and you damage those muscle cells, they start dividing and they get bigger. They're not getting bigger because each cell's getting bigger, but you're actually increasing the number of cells they're dividing. And then some cells are temporarily in G0 for the checkpoint and for DNA repair, because we can repair some minor damage to our DNA. However, if it's determined during that checkpoint that the DNA damage cannot be repaired, What's going on here? Oh my goodness, sorry, there's a really big lag right now. I don't know what's happening. Help, technology. I miss my whiteboard. If DNA cannot be repaired, cell must be destroyed. You do not want bad cells dividing. When bad cells are allowed to divide, you get cancer. There are some tumor suppressor proteins that are part of this checkpoint. Oh my gosh, it's not often that I spell things wrong, but I think I'm spelling suppressor wrong. No, that's right. It's 2 P's and 2 S's. That just looked weird to me. Sorry. Tumor suppressor proteins are part of the checkpoint, and they're going to signal the immune system to destroy that cell. That's why they're called tumor suppressor. They're keeping malignant tumors, cancerous tumors from developing. So if a cell has bad DNA, they're going to signal that cell to go into apoptosis, or you're going to have some T cells that destroy that cell and not allow a bad cell to divide. And that's all regulated by these important tumor suppressor proteins. We're learning more and more about how things can go wrong in the cell cycle and what causes cancer. And if you have a DNA mutation that causes your tumor suppressor proteins to not function correctly, you have a high likelihood of getting cancer during your lifetime. So we're learning more and more about the importance of these tumor suppressor proteins in controlling cancer. We'll talk more about those toward the end of this lecture. If the cell, though, can either be repaired or there wasn't any DNA damage, then that clock continues. So after that G1 checkpoint, if everything's good, then the cell cycle starts again. So it goes back into G1, continues through S, the DNA is copied, goes into G2. And in G2, it's mostly doing its cell job, but it's also getting ready to divide. Getting ready to go through mitosis. What kinds of things would it be doing to get ready to divide? Well, it would be making more organelles, because remember that cell needs to divide everything in half. So if it needs a lot of something to start its cell job, it's going to really make a few more right now. And it's also going to replicate its centriole. Remember, the centriole is the structure that's going to organize those microtubules to pull those sister chromatids apart during cell division. And so each cell only got one when cell division happened. So during the previous cell division, each new cell only got one centriole. So in G2, as part of that getting ready to divide, the centriole replicates. So you go from 1 to 2 centrioles, because we need one to migrate to each end of the cell so that we have microtubules coming from each end of the cell to pull those microtubules apart. Sorry, I'll just keep that there for a second. Going back to our clock of the cell cycle. This is where I really miss my whiteboard, so I can just flip to a board instead of having to scroll all the way up to this picture. To make sure everything is ready to roll, there's another checkpoint that occurs here called the G2 checkpoint. And it's really checking to make sure that centriole replicated. and that this cell is ready to divide. So G2 checkpoint. Did centriole replicate? Is cell ready to divide? And in most cases it is. This isn't a major checkpoint. The G1 checkpoint is really the big, most important checkpoint. If everything's a go, now this cell is going to move into mitosis, which is where the cell is going to divide. Mitosis is actually divided now into 5 stages, but it used to be 4. And if you were in lab, we would only be observing 4. So we're going to be a little old school here and divide this into what's called PMAT. Prophase, metaphase, anaphase, telophase are all part of mitosis. And we're now going to talk about what happens during each of those stages of mitosis. Remember, interphase was G1, S, and G2, and the DNA is uncondensed. So it's just all chromatin. Mitosis is going to include prophase, most textbooks now have prophase broken into prophase and prometaphase, but we're going to include what happens in prophase and prometaphase together. We're going to consider these the same. Prometaphase was added at some point in biology. We're going to call it all prophase. Metaphase, anaphase, telophase. I'm going to, while I'm talking to you, I'm going to tell you when prophase really becomes prometaphase, but again, we're going to call this all prophase. So DNA is uncondensed. We're in G2. You get past that G2 checkpoint, and now that cell enters prophase. Remember that in interphase, nucleus, DNA is all stretched out, uncondensed, and now that needs to get more organized. The goal is to pull the two sister chromatids apart, and in order to do that, there needs to be more organization. It can't just be all tangled up. So as that cell moves into prophase, one of the first things that's going to happen is that that DNA is going to condense and become visible as individual chromosomes. And the nuclear envelope is going to start breaking down, because for those microtubules to get in there and pull those sister chromatids apart, you can't have a nuclear envelope. And microtubules are going to have to start emerging from the centrioles that have migrated to opposite poles of the cell. And eventually those microtubules need to grab on to the chromosomes, specifically with each sister chromatid attached to a microtubule from the opposite pole. In reality, these chromosomes are obviously aren't different colors, but I'm going to draw them different colors so you can remember a couple of things. One is that one of these chromosomes came from mom and one came from dad. And that's not really important in the mitosis story, but we need to be able to compare meiosis and mitosis. So it is going to be important at some point. So I'm going to draw them two separate colors. Everything that came from dad is going to be blue. So here's a chromosome number 1 from dad, and everything from mom is going to be red. This is a chromosome number 1 from mom. And they wouldn't necessarily be next to each other like that. Here's a slightly smaller one from dad, and here's that same one from mom. So here's what's happening during prophase that you need to know about. DNA condenses. And if you were looking under the microscope, you would see individual chromosomes being visible for the first time. It's wrapping around those protein beads called histones, and now it's visible. It's this tightly packaged, organized little package of DNA now, much easier to pull apart. Centrioles migrate to opposite ends of the cell. Microtubules emerge from the centrioles. Nuclear envelope fragments and eventually disappears as this progresses. And now microtubules attach to kinetochores on each sister chromatid and realize that each sister chromatid is attached to a microtubule from an opposite pole. So typically, if we were calling this metaphase, prophase, prometaphase, and then metaphase, right here, where the nuclear envelope breaks down and those microtubules attach, this would be prometaphase. But we're calling this all prophase. I don't really know where to write that. We're calling that all prophase. But if you need to know prometaphase for the future, when that nuclear envelope disappears is when you're in prometaphase. So now going back to our picture for a second, microtubules have grown and these sister chromatids are going to be attached to opposite poles. And realize that those microtubules are connecting to a kinetochore. And there are also going to be some microtubules that don't connect. And those are going to just be connecting to each other and elongating that cell. So the ones that are connected are called kinetochore microtubules. And then you have non-kinetochore microtubules. I don't want you to know those terms. If you go on to take Bio 200 or maybe even in physiology, they differentiate between those microtubules. Just realize not all of them attach to sister chromatids, but the ones that do are really, really important. So now imagine that each of those chromosomes is getting pulled from those microtubules at opposite ends. And as they pull, everything is going to get pulled until it aligns in the middle. And when everything aligns in the middle, we're at metaphase. At metaphase, obviously there's no nuclear envelope anymore. And now those chromosomes are aligned in the middle of the cell. And it's completely random how they align. It could be because it doesn't matter. The two number ones don't need to be together and the two number twos don't need to be together. And the reason for that is the goal is just to pull those sister chromatids into opposite ends of the cell. Realizing that these are attached to kinetochores, but I don't want to take the time to draw that on every single one of these every time. But they've been pulled, and now they're all in the middle of the cell. This imaginary middle of the cell, sometimes called the equator of the cell, is called the metaphase plate. And this is where the chromosomes are all aligned with each sister chromatid kind of facing the opposite ends of the cell. Sometimes the pictures will be flipped sideways when these are viewed. And so the chromosomes will be aligned a different direction. I like drawing them this way. It's kind of both ways, depending on which kind of textbook you look at. Also, if you look at these cells under the microscope, sometimes they're mounted in a different orientation as well. But this is when another really important checkpoint takes place called the M checkpoint. And it's making sure that a microtubule is correctly attached to each sister chromatid. So are the microtubules correctly attached? To each kinetochore. Imagine if that one wasn't. Now what's going to happen is when this divides into two new cells, one cell's not going to get any of that chromosome and one's going to get too much. So one's going to get too much DNA and one's going to be missing some DNA. So that would be an error in cell division. That checkpoint is making sure that everything is correctly attached to a kinetochore. And of course, then there are these non-kinetochore ones that are elongating the cell, because eventually we want to make two new cells. So it needs to keep elongating the cell. If everything checks out and everything is considered to be correctly attached, then the signal is given for those two sister chromatids to be pulled apart. Those sister chromatids are being held together with some sticky proteins called cohesin proteins. Not cohesion, but cohesin proteins. So these are some sticky proteins that hold the sister chromatids together. And when that M checkpoint is finished, if everything is correctly connected, the signal is given for an enzyme called Suppressor. I love this enzyme name. Suppressor, we know it's an enzyme because it ends in ASE, and it's going to cause the sister chromatids to separate. This enzyme called Suppressor hydrolyzes or digests the cohesin proteins, and the sister chromatids separate. And the second those sister chromatids separate, that cell is now in anaphase. In anaphase, sister chromatids separate and migrate to opposite ends of the pole. Or sorry, opposite ends of the cell, opposite poles of the cell, I should say. Trying to remember which one I drew first. Red, red, blue, blue. Just trying to stay a little consistent here. One identical piece of DNA to each end of the cell. Okay, so in anaphase, sister chromatids separate. And those microtubules actually start shortening, and that causes them to move to opposite ends of the cell. Also, those non-sister, I'm sorry, I'm saying the wrong thing. those non-kinetochore microtubules have really elongated the cell now. I'm not really drawing it that way, but it's really long now. And in this last stage, telophase, this can be pronounced telophase or telophase. That's an E. Two important things happen. One is that new nuclear envelopes form around those separated chromatids. So new nuclear envelopes form. And now that's effectively division of the nucleus of the cell. Now the cytoplasm has to divide. And this division of the cytoplasm is really what causes us to have two new cells. And it's called cyto, which means cell, cytokinesis. If this is a plant cell, the way that cytoplasm is going to divide is by just laying down new cell wall and plasma membrane between the two. And it's going to just kind of be like this brickwork where the cells are going to all just be stacked on top of each other. And eventually those will grow into full-size cells. So cell plate in plants is how cytokinesis is achieved. If this was an animal cell, it would actually pinch the cytoplasm in half. And this indentation that happens while that cell is dividing is called a cleavage furrow. So cytoplasm is pinched in half in animal cells. So they end up being completely separate from each other. It's a ring of actin, which is contractile, that pinches those in half. And now we end up with two new genetically identical cells. But see, each chromosome has one chromatid. Now, when each of those two new baby cells goes through the cell cycle, it will go through the S phase, and that DNA will get replicated, and that cell can go on and divide itself. So telophase gave us two new genetically identical cells. This is probably a good time to pause the video and take a break. It's been a lot so far. We still need to talk about meiosis and what can go wrong in cell division. I'm not going to talk about meiosis in as much detail because a lot of the steps are pretty similar. So I'm going to rush through them a little faster. And then I'll show you them again when I show you the slides. Remember, meiosis is division of a 2N cell to produce 4 genetically different N cells. In other words, a diploid cell is going to divide twice and produce 4 genetically identical, 4 genetically different, not identical, the opposite of that, 4 genetically different haploid cells. Two divisions They're called Meiosis One and Meiosis Two. Meiosis One includes Prophase One, Metaphase One, Anaphase One, and Telophase One. And then same thing for Meiosis Two. I'm just going to write PMAT this time because you now know what that stands for. Prophase 2, Metaphase 2, Anaphase 2, and Telophase 2. The basic setup is the same. What differs is what is being separated. But you're going to see when I tell this story, a lot of the story is the same as mitosis and what happens in each of those stages. There are some really important differences though, because obviously this is going to be a reduction division and we're ending up with genetically different cells. Remember, this is a cell that has 46 chromosomes in humans and we're getting it down to 23. As this cell enters Meiosis One, Prophase One, there's something really, really important that happens. I'm going to tell you first, and then I'll attempt my bad drawing on my iPad. In Prophase One, something pretty amazing happens, and that is homologous chromosomes come together synapsis and crossing over occur. Remember, the homologous chromosomes are the same number and they code for the same traits in the same location. So let me talk a little bit more about that. You have 23 pairs of chromosomes. And if we looked at a picture of your chromosomes, you would see that for chromosome number 1, you have one of those that came from mom and one of those that came from dad. And for chromosome number 2, you have one of those that came from mom and one of those that came from dad. Same for 3. Oops. And so on, all the way to number 23. Chromosome number 23, these are your sex chromosomes. Numbers 1 through 22, those are what are called your autosomes. This is an important distinction, especially when we start talking about heredity and how we inherit certain traits. This difference between 1 through 22 and 23. We all have the same 1 through 22, whether we're male or female. This is where we differ, is in the sex chromosomes. If you got an X from one parent and an X from the other, you're genetically female. And if you got an X from mom, but you got a Y from dad, you're genetically male. There are variations of that we will talk about. But if meiosis occurred correctly, then you got either 2 X's or an X and a Y. Those are called your sex chromosomes. Those are not the same thing as gametes. Okay, students get these terms confused all the time. Your sex chromosomes are your 23rd pair of chromosomes. Let's say this is an egg, which is a gamete. In this egg are chromosomes 1 through 22, which are the autosomes, one of each, and a chromosome number 23, which is the sex chromosome. Okay, same thing in the sperm. One of each for 1 through 22, and then there's a 23rd. Females can only give their offspring an X because they only have X's. So 50% of their offspring are going to get this X and 50% are going to get this one. In males, when they go through this reduction division, 50% of their sperm get this X and 50% are going to get that Y. So males can give an X to some of their offspring and a Y to others. Females can only give X's. So it's really the male, the sperm, that determines the gender of the offspring. So sex chromosomes are not the same thing as gametes. Because you have two of each, they are called homologous. So your two number ones are homologous chromosomes. Your two number twos are homologous chromosomes. Your two number threes are homologous chromosomes. So homologous chromosomes have the same chromosome number. And the reason they have the same chromosome number is they code for the same traits in the exact same locations. Every human on Earth, if there's a specific gene, it's in the same place on the same chromosome. So if it's the gene that codes for a lactase enzyme, every single one of us has that gene on the same chromosome in the same location. These two homologous ones, one came from mom and one came from dad, but they code for the same traits in the same location. How they code for it is a little bit different because they're not identical twins, obviously, but they're called homologous. I love this term, homologous. Same language. These two chromosomes are speaking the same language. They're telling the cell to make a protein in this location. They're speaking the same language. They code for the same proteins. Sorry, my throat's getting really dry. I know I'm out of water, so I might have to pause in a minute and go get some more. During prophase 1 of meiosis, homologous chromosomes come together. This does not happen in mitosis. Remember, in mitosis, the whole goal is just pull sister chromatids apart. So it doesn't matter where the two number ones are. They could happen to be next to each other just by accident, but they don't need to be, okay? Because we're just pulling the two identical pieces of DNA apart. But now something amazing is going to happen. So the two number ones get together, the two number twos, and so on. And let's say this is chromosome number 1. So here's one from dad. And here's one from mom. And they're actually going to physically touch each other, which is called synapsis. And so these are homologous chromosomes. So this is chromosome number 1. And we are in prophase 1 of meiosis. So homologous chromosomes come together. Synapsis occurs. And crossing over. Crossing over is gene swapping between the chromosomes. Sorry, that word chromosome is a little squished there. This is the first way we're going to get genetic diversity. So remember, this chromosome number 1 came from dad, and this chromosome number 1 came from mom. By the time we're done, those are going to be a mixture of DNA. That is crazy. To me, this is one of the most amazing things that happens in biology. Those chromosomes are going to swap traits. So think about it right now. You're making eggs or sperm, and you have DNA from your mom, and you have DNA from your dad. And when you make eggs and sperm, they're not purely one parent's DNA or the others. They're going to be this mixed up hodgepodge of your 2 parents' DNA. So your babies end up being a blending of your parents and your partner's parents. At the end of prophase 1, the number of genes that got swapped is totally random. But here's the deal. It's going to happen in several different points on these chromosomes. So at the end, they're not going to be purely one parent's DNA or the other. And I'll tell you right now, these 4 chromatids, when these are together, this is called a tetrad. Tetra is 4. These 4 chromosomes, 4 chromatids on 2 different chromosomes, those are going to end up in 4 different cells. Imagine that happening for 23 pairs of chromosomes. And you can start to see how every egg or sperm is going to get different DNA, a different combination, a different blending of DNA from this individual's parents. That's pretty crazy. The goal of Meiosis One is then to pull this homologous pair of chromosomes apart. And that's what's going to happen in Meiosis One. So Meiosis One separates homologous chromosomes completely intact. Sister chromosomes don't get pulled apart at this point. So the way the microtubules are going to attach is one microtubule attached to each homologous chromosome. And I'm going to show you a picture of this. I'm not going to take the time to draw this. I'm going to show you some slides in a minute to show you a picture. But imagine pull, 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 and now the 2 homologous chromosomes end up in the center of the cell with each homologous chromosome facing opposite poles. They get pulled apart and they end up in 2 separate cells. So just very quick drawing. Prophase. I'm just going to draw 1 pair of chromosomes here. They paired up, they did some gene swapping, and so now they're kind of a mixture of DNA. Microtubules attach so that each homologous chromosome is facing the opposite pole. So now in Metaphase One, we've got this situation. Oops. And then at Anaphase One, the sister chromatids are staying intact, but that whole homologous chromosome is going to get pulled to the opposite end, fully intact, like this. Sorry, I want to take the time to make these a mixture of DNA each time. And now this is going to divide into 2 separate cells in Telophase One. So prophase, metaphase, anaphase, telophase. I'm just writing that first letter. nuclear envelopes reform in some organisms, and some they don't reform because it's just going to move right into prophase 1 again. But these divide into 2 separate cells. And now each cell has a fully intact, but just one. So we're already at the haploid number of chromosomes. And they're already a mixture of the 2 parents' DNA. Okay, so here, this cell has 2 chromosomes, and each of these just have 1. So we've already reduced the amount of DNA in half. But the sister chromatids are still attached, and meiosis 2 separates sister chromatids. And it looks just like mitosis. The difference is you're starting off with half the number of chromosomes. Before I show you that, though, I want to point out something really important, and that is there's another way that genetic diversity occurs, and that is during Metaphase 1, right here. The way that pair flip-flops on the metaphase plate, remember metaphase plate is right here in the middle of the cell, it's completely random. Okay, so remember this blue one was originally from dad, and this one was originally from mom. And now they're a mixed up hodgepodge, but the way those 4 chromatids looks is completely different. And the way that pair flip-flops on the metaphase plate, how it ends up is totally random. Could be, this way too. And that's called independent assortment. This is the second way that genetic diversity occurs in the gametes. Remember, the first way was crossing over during prophase 1. This independent assortment is random alignment of the homologous pair. During metaphase 1. And it's random alignment on the metaphase plate. And it leads to genetic diversity because it's going to be different every time how these flip-flop. And that's how you end up with some gametes that have more of mom's DNA and some that have more of dad's DNA, just because of how they flip-flop on that metaphase plate. And that's going to affect the genetics of the offspring. Okay, I'm going to pause this recording for a minute, but I will be back. I need to get some more water and I need to plug in my iPad. Okay, I'm back with water and my iPad's plugged in. Hopefully my Apple Pencil won't die now. It's a long lecture. Okay, independent assortment is how we're going to get genetic diversity. Remember, plus crossing over, which was the gene swapping in prophase 1. Now in prophase 2, that doesn't happen because remember the homologous chromosomes have been separated. They're in 2 different cells. There is no way for crossing over to occur. There's no way for homologous chromosomes to get back together. That ship has sailed. They're in 2 different cells now. That can't happen. So as we go through prophase 2, metaphase 2, anaphase 2, and telophase 2, those homologous chromosomes are in 2 separate cells. Remember that those 2 cells each just have 1 fully intact chromosome. Getting messier and messier with these as time goes on. And of course, they're a blending. I don't even know if that's how I originally drew them, but they're all 4 different, right? All 4 of those chromatids are different because they're a blending of DNA from the 2 parents. So as we start prophase 1, there is no crossing over that occurs. There is no pairing up of homologous chromosomes. They're in 2 separate cells. But the goal is to pull the sister chromatids apart. So that's how the microtubules are going to attach. And they're going to pull so that at metaphase, this is prophase 2. So that at metaphase 2, they're aligned in the middle. I've kind of already shown that right there. They're already practically aligned in that first picture. But they would be perfectly aligned in the middle. And then at Anaphase 1, Anaphase 2, we're in 2, Kim. I'm trying to take shortcuts here and it's really just creating more work for me. In Anaphase 2, sister chromatids separate. I'm already realizing I didn't leave enough room for the rest of the story here. Those are going to divide into become 2 separate cells, which is going to make 4 because each of those is going to split in half. So in telophase 2, see if I can squeeze it in here. Those are going to separate. Cytokinesis and new nuclear envelopes form. And each one of these chromatids is now in a different cell. And what used to be just mom's DNA and just dad's DNA is now a mixture. And you can see that there are now 4 haploid cells. So they have half the number of chromosomes that we started out with. So we started off with 2 chromosomes and each of those cells just has 1 chromosome and they're all 4 genetically different. And I just drew 1 chromosome. Imagine, well, I drew 1 pair of chromosomes. Imagine 23 pairs of chromosomes, how much genetic variation you can get in those gametes. Genetic variation in a population is critical to survival of that population long term. Imagine if everyone was genetically the same in a population of organisms. I mean, just think it's a population of rabbits and there was no genetic variation. They were all genetically identical to each other. A virus, a fungus, a bacterial infection, changes in climate, so, just changes in temperature, changes in air quality, changes in water availability. If everyone's genetically the same, there's no variation to withstand changes in environmental conditions. Without that genetic variation, nobody survives. The reason that we as humans are still here today after this many thousands of years is because of our genetic variation. And even though we're all pretty similar to each other, that small amount of variation means that somebody survives. So not everyone is killed by certain viruses, certain bacteria. Not everyone is killed by changes in the climate right now because we have that genetic variation. I mean, things that you don't even think about, like there are a lot of studies about how most humans who still survive have some tolerance to alcohol, because there was a time in history where alcohol was a clean source of drinking water and people didn't realize that you couldn't just drink as much of it as you wanted. And so a lot of people died from it. And those who are remaining are those who can metabolize alcohol. Just crazy things like that that you don't even think about. So Natural selection was something we learned about in the very first week of class, and that's because it's so important in understanding almost everything about biology. If there's not that genetic variation, then natural selection, there's no variation to act on, right? Everybody gets wiped out. And that's what we as humans have done on this planet for certain species of animals and plants and fungi and protists. And we have really taken those populations down to such small numbers that there's not that genetic variation anymore. And so let's think about just in California in our history that elephant seals were hunted to near extinction. And now there are several thousand of them on the beaches in California, but because they were hunted to near extinction, that very small population recovered has very little genetic variation. So even though the numbers are there, the diversity isn't, and the fear is that some virus or other situation could come along and wipe out everyone because that variation is not there. So this genetic variation in the gametes is very critical to survival of populations. That variation ensures that there will be some individuals who survive almost every situation that the environment can throw at them. Things can go wrong in meiosis and mitosis. And so I want to talk about errors in mitosis versus meiosis. Let's start with mitosis. We have a cell with 46 chromosomes. Let's say it's a skin cell. And it goes through the cell cycle, and something goes wrong. And when it divides, one cell ends up with 45 chromosomes, and one ends up with 47. So one is missing a chromosome and one has an extra. This usually occurs due to something called non-disjunction, which is failure of the chromosomes or chromatids, depending if we're talking about meiosis or mitosis and which stage, to separate correctly during cell division. And that results in the wrong number of chromosomes. If this is a skin cell, a stomach cell, whatever the case might be during mitosis, this is not a big deal. First of all, you have thousands of the same cell type. So even if these two have the wrong number of chromosomes, it's not totally critical. Sorry, for some reason my iPad quit showing my screen. I have to pause for a second. I'm back, sorry about that. It's a skin cell or another body cell. You have thousands of the same type. So the fact that these two aren't making proteins correctly for those chromosomes that are wrong, they're the wrong number of, is not that critical. Also remember, there's that G1S checkpoint that is going to detect that they're the wrong number of chromosomes, and these cells will be destroyed and they will not be allowed to produce two more with 45 or 47. So cell cycle checkpoint is going to take care of that problem. And also you have thousands of the same that are probably doing the job right. So the fact that these two aren't doing the job right is not that critical because there are a lot of others that are making proteins correctly. That is not the case in meiosis though. Remember, meiosis is making gametes. And if there's an error in meiosis, you start with a cell that has 46 and you go through 2 rounds of division and you end up with 2 that have 22 and 2 that have 24. Remember, normal number is 23. Here's the problem. Let's say one of these with 20, let's say this is an egg. and it has 22. It's missing a chromosome. And it's fertilized by a sperm with 23. That's going to lead to a zygote with only 45. So that zygote is missing a chromosome. Remember that now we're not talking about one cell once this thing divides. That zygote is going to divide, and now every cell of this organism has the wrong number of chromosomes. Obviously, the one with too much DNA, if that was fertilized by one with 23, that's going to lead to a zygote with 47. And this one has too many chromosomes. It has an extra. So that karyotype that I'm going to show you, I'm thinking of when I recorded using the slides. I'm going to show you a karyotype in a minute that shows you an extra chromosome number 21. And having an extra chromosome in every cell of your whole body is devastating. So an error in meiosis is a big deal. Error in meiosis equals every cell has the wrong number, not just two cells. And there's no way to fix this. It's every cell of the entire organism that has the wrong number. If you have 3 of a chromosome, it's called a trisomy. And if you only have 1 of a chromosome, it's called a monosomy. Somy means body. So 3 chromosome bodies or 1 chromosome body. Remember, you need 2 of every chromosome to make your proteins correctly. You have to have 2 of every chromosome to make your proteins correctly. For the autosomes, here's the rule. There are no viable, meaning survivable, and this is in humans, there are no viable autosomal monosomies. You must have at least 2 of a chromosome. Remember, autosomes are 1 through 22 on your chromosomes. You can't just have 1 of any of your autosomes. You must have at least 2 of every chromosome to develop. There are no viable no viable autosomal monosomies. My brain is getting tired. Autosomes are 1 through 22. So if something's called autosomal, it's referring to chromosomes 1 through 22. We'll be talking a lot about autosomal inheritance when we talk about patterns of inheritance next week. There are some trisomies. There are a few autosomal trisomies that are viable, but most of them don't develop. Obviously, trisomy 21 is really the only one that reaches adult age. All the others are lethal. It's too many genes that are messed up. It just so happens that 21 is our smallest chromosome. It codes for the smallest number of proteins, and those proteins aren't critical for survival. So trisomy 21 is Down syndrome, and those individuals can become adults. It's the only autosomal trisomy that reaches adulthood. There are sex chromosome trisomies that are viable. You can be XXX, XXY, YYY, And you might not even know that someone is that. There's one sex chromosome monosomy. So this is the only monosomy in all of biology. Why is my iPad doing this? I'm just going to show you on the board because I'm about to switch to my slides anyway. So, okay, you can have just one X. And that person's genotype is usually written as XO. It means from one parent, they didn't get a sex chromosome. So they're missing a chromosome number 23. This is called Turner syndrome. Turner syndrome. Oh my goodness. I want to be in the classroom with my boards. And Turner syndrome means you just have one X. So this person is genetically female and has some issues, but this person becomes an adult. And I've had a student with Turner syndrome before. So there is one viable sex chromosome monosomy, but no autosomal monosomies. Okay, so non-disjunction, failure of the chromosomes to separate correctly. That can happen in mitosis or meiosis, far more devastating in meiosis for the reasons that I already outlined. Okay, I'm now switching to my slides. I just want to quickly review some slides with you. So let me share my screen. I'm going to pause again while I share my screen with you. So just a couple of review slides before we get to the new material at the end. I just want to review because I drew some pretty bad diagrams on my iPad, and iPad's going in and out and the whole thing. So just a reminder, somatic cells are every cell of the body except for eggs and sperm. Gametes are the eggs and sperm. Diploid cells would have the double number of chromosomes. Haploid have half the number. So you can see in this one, the diploid number was 4 chromosomes. That means the haploid number is 2 chromosomes. So this would be a somatic cell, and this would be a gamete. The goal of mitosis is to start with a cell that has the diploid number and end up with 2 genetically identical diploid cells. The goal of meiosis is start with a diploid cell, but end up with 4 haploid cells. And those 4 haploid cells are all genetically different from each other. The reason cells divide, go ahead and type those in here as a review. Development, so when that original zygote developed into a whole organism, that required a lot of cell division. And then that organism has to grow into an adult organism. Replacement of cells that have been damaged and repair of damaged cells. I'm really doing great here. And then, of course, that different type of division, reduction division called meiosis for production of gametes. This is a human karyotype. It's a picture of your chromosomes, and you can see 23 pairs of chromosomes. 1 through 22 are the autosomes. So these are homologous chromosomes, homologous chromosomes, homologous chromosomes, same number. They speak the same language. They code for the same traits in the same location. Number 23, these are the sex chromosomes. This individual has an X and a Y, so they are genetically male. Sex chromosomes are within each gamete. So those are 2 different things. Here's a trisomy 21 karyotype. This individual is female. They have 2 X chromosomes and no Y, and they have an extra chromosome 21 in every cell of their bodies. This happened due to non-disjunction during meiosis. So either the egg or the sperm, most likely the egg, because the chances of non-disjunction in the eggs increase with age. The reason for this is, girls, we are all born with all of our egg cells through Meiosis One and just resting in our ovaries. Every 30 days, Does that timeline sound familiar? About every 30 days, one of those egg cells goes through meiosis too. And so by the time you're 40 years old, those egg cells have been sitting in your ovaries at Meiosis One for over 40 years. And the chances of an error during meiosis increase with age. We don't have statistics for how many trisomies happen in chromosome number 1 or chromosome number 2 or some of these bigger chromosomes, because this baby never develops. A trisomy of this critical chromosome that codes for so many proteins. You can see how big this chromosome is. It codes for so many traits. That's too many proteins that are being made incorrectly, and that baby never develops, and we don't have stats for that. We only know the ones that result in a birth or develops to a certain point and genetic testing is done. And obviously this individual could reach adulthood because look how tiny that chromosome is, much smaller than chromosome number 1. And it just so happens that those proteins aren't critical for survival. Again, we can have a lot of variation in the sex chromosomes. It's really the autosomes that are crucial. You can't just have one of the autosomes and develop. This whole thing's a chromosome. Each one of these pieces of DNA is called a chromatid. If they're attached and identical, then they're called a sister chromatid. This shows DNA condensing, wrapping around. So here's the DNA in its double helix. Starts wrapping around those histone beads and becomes coiled, 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 and ends up being that fully condensed chromosome. Remember, when it's all stretched out, it's called chromatin. And that's how the cells look in interphase when they're doing their cell job, mostly making proteins. The centromere is this constricted region of the cell. So this is all review, that's why I'm going fast. The kinetochore is where those microtubules attach. Here's the clock of the cell cycle, G1, mostly doing its cell job. DNA replication at the S phase, getting ready for cell division in G2, but also just doing its cell job. Then the 4 major stages of mitosis: prophase, metaphase, anaphase, and telophase. The cell cycle checkpoints, we talked about G1 mostly being to check for DNA damage. You can see there are some other things that are checked there, but I don't need you to know that full list. G2, really, it doesn't even list what I told you, and that is that centriole has to replicate. So we make sure that microtubules are going to grow from each end of the cell and attach to those sister chromatids during mitosis, or homologous chromosomes and sister chromatids during meiosis. And then there's that M checkpoint to make sure everything connected correctly. Remember, G1 checkpoint, if that cell's damage cannot be repaired, then that cell needs to be destroyed. And when something goes wrong and bad cells are allowed to divide, we get cancer. In that G0 phase, it could be because that cell is a cell that will never divide again. So this is a nerve cell in the brain or spinal cord, and it's irreversible for this to go back into G1 or S or G2. This cell is just quietly sitting there, or maybe it's going through DNA repair and it can go back in. So this shows that a cell goes into G0 and then it goes back into the cell cycle. So that would be a cell that's being checked. It's going through a cell cycle checkpoint, or maybe it's going through some DNA repair. The stages of mitosis: interphase, no visible chromosomes, it's just chromatin. The DNA condenses, that nuclear envelope fragments and disappears. And those microtubules attach. They're pulled when everything's in the middle of the cell. That's metaphase. When the chromatids separate and move to opposite poles, that's anaphase. And in telophase, nuclear envelopes reform. The cytoplasm divides through cytokinesis, and you end up with 2 separate genetically identical cells. I just wanted to show you some cells at each stage here. So these cells where you can't really see anything, really you can't see any individual chromosomes, those are all at interphase. And I'll just go ahead and show you on this same slide. Let me just make this bigger. See where it starts looking a little wormy? That's what I always say in class when we look at these under the microscope. This shows you that those individual chromosomes are starting to condense and become visible. So this cell is moving into prophase. In fact, you can see instead of having this nice round nucleus, this one has a nuclear envelope still. This one has a nice intact nuclear envelope. You can see it's starting to lose its shape as that nuclear envelope breaks down. Everything pulls once it gets connected to the microtubules. And here you can see microtubules. And right here in the center of the cell, you can see alignment at the metaphase plate. So that would be metaphase. And then they're going to pull apart in anaphase. Check this out. How cool is that? So here, visible, aligned in the middle, and now the chromosomes have pulled apart. So realize this is an onion root, and every single one of these cells in the onion root is at a different stage of cell division. So your cells don't all divide at the same time. They're all on their own clock on that cell cycle. And these are all at a different stage. Now, most of them are at interphase. Interphase. All these are at interphase. Only a couple of these are dividing. And then finally, at telophase, those cells are going to divide. This is the cell plate, which is separating those two cells, and they'll eventually grow into the same size as these cells above and below them. And then this would be an animal, that cleavage furrow. That cytoplasm is pinching in half to form two new cells. I wanted to show you here cohesin proteins holding those two chromatids together. Kinetochore is attached to microtubules. And you can see here, cohesin proteins are what is holding those chromosomes together. And when they separate, that's anaphase. Meiosis is going to result in 4 genetically different cells. And I just wanted to show you, this is, well, I don't want to give you a different term. This term chiasmata, that's the spot at which synapsis and crossing over is occurring. But here's that gene swapping happening during prophase 1. So synapsis is really them coming in contact with each other, and then crossing over is the gene swapping. And this slide kind of tells you that. So synapsis refers to the pairing of the chromosomes, whereas crossing over is the exchange of genetic material. Doesn't always happen. Sometimes two chromosomes can come together and they don't swap genetic material. That's another way that it's random each time. Also, just pointing out independent assortment. So you can see that depending on the alignment of how that pair flip-flops on the metaphase plate, that you're going to end up with a different outcome. So you can see here, it's just one pair, this one in the middle. Right here, it's red on the left and blue on the right. And here it's reversed. And you're going to end up with different outcomes based on that. So the 2 ways that genetic diversity occurs in the gametes, crossing over during prophase 1 and independent assortment during metaphase 1. Random alignment on that metaphase plate. So at the end of telophase 1 of meiosis, the chromosome number is reduced, and really it's that fully intact homologous chromosome. So telophase 1 is going to really completely separate those homologous chromosomes into 2 different cells. And then in meiosis 2, it's very similar to mitosis in that the sister chromatids get pulled apart. I'm going to post these slides so that if you want to study them in more detail, you can do that. So again, if you look at what we started with in Meiosis One, that pair separates completely intact into two different cells. The only thing this isn't showing is it's showing one completely red and one completely blue. But realize it would now be a mixture of those two, so they'd be stripy, a combination of those two colors. But you can see then sister chromatids separate in meiosis 2. And so you end up with 4 haploid cells. By the way, these will each go through the S phase of the cell cycle, and that chromatid will replicate, so they'll end up looking like this again. This is a really good slide to just really study the stages of meiosis, and I'll post this for you. Again, errors in meiosis are far more damaging than in mitosis. I want to just talk quickly about cancer. And these slides have a lot of words on them purposely because I really want you to get all of this information. So cancers derive from a single cell or more than one, but it's from single cells, not all of the cells of your body, that have acquired characteristics to cause them to keep dividing out of control. In other words, in a cell, something goes wrong in the cell cycle. Those checkpoints aren't working. The tumor suppressor proteins aren't doing their job, and they divide in an uncontrolled, unrestrained manner and eventually start invading the surrounding tissues. They also recruit blood flow to them and take blood flow away from the surrounding tissue. So those are the ways that it damages the organs and tissues in your body. You might think, big deal, why is it a big deal that a tumor is growing there? Well, it's a big deal because it's taking the blood supply from your healthy organ, recruiting that blood flow to the tumor, and it's also damaging and killing the tissue of that organ. They behave this way because of changes in the DNA of those cancer cells. Those genes that are changed are now called cancer genes once that change starts because they cause cancer. So these are genes that used to be normal, healthy, functioning genes. Remember, a gene is a region of DNA that codes for one protein. 1 in 3 people in the Western world develop cancer, and 1 in 5 die from cancer. 20% of the population will die from cancer. That could be even higher within your lifetime. There are approximately 200 types of cancer. Each has different causes, symptoms, and treatments. And your risk of developing cancer usually depends on your age, but also your lifestyle and definitely your DNA. Okay, we all have seen people who are 90 years old and they're still smoking a cigarette every day. They have really good tumor suppressor genes. Their tumor suppressor proteins are working correctly. They also have good DNA repair mechanisms. So DNA plays a huge role. You almost have to have the perfect storm. Just because you're genetically predisposed for a certain cancer, if you're not exposed to certain things and you don't develop that high risk, then maybe you'll never develop that cancer. It kind of has to be everything coming together for that cancer to happen. There are 2 types of mutations that can happen. One is just in your somatic cells, and you obviously can't pass that on to your offspring. It's when a change happens in your eggs or sperm that you can pass that on to your offspring. So a change that happens in eggs or sperm, you inherited that from your parents, and your kids could inherit that from you if you have that same mutation in your eggs and sperm, which if you got it from your parents, then you do have it in your eggs and sperm. So sometimes they can be inherited, but sometimes they can be acquired during your lifetime. And believe it or not, inherited cancers are this yellow, and they're not as common as just changes to your DNA that happen during your lifetime because of things you're exposed to. Toxins in the environment, drugs, alcohol, pollution, stress. Lots of things that we don't have control over, pesticides in our food, plastic bottles, things like that, all of these chemicals in our world that can cause changes to your cells during your lifetime. So most of these mutations are acquired. Okay, so when cancer cells are growing, they're not following the same rules that normal cells follow. There are signals that tell our cells when to stop growing. For example, think about it. You cut your hand and now there's this space that needs to be filled in, and your cells know when to quit growing. When they come in contact with each other, they don't start growing in this giant mound on your hand. Cancer cells that form tumors, they just keep growing. They keep dividing indefinitely. They're immortal. They can't follow the same rules anymore as a normal cell. These are some external factors that can cause cancer: smoking, environmental toxins, radiation, viruses. So human papillomavirus, HPV, causes cervical cancer. That's what the Gardasil vaccine is for. The first vaccine ever developed to completely prevent cancer. That's pretty amazing. And we're learning more and more about how our lifestyle affects our cancer risk. But you can inherit mutations in your DNA. You could also inherit hormone disorders that can result in cancer, immune conditions that can make you more prone to cancer if your immune system isn't doing its job in taking out those defective cells. And then you can have just random mutations during your lifetime that can occur regardless of whether you're exposed to something or not. So a lot of these things can happen together at the same time or just individually, but they will result in an abnormal cell behavior and excessive cell division. It's when the cell division gets out of control that it's a bad thing. Those masses grow, they expand, they surround, and really affect the normal tissue. And if they can break off and move to other areas, they're called metastatic. So metastasis happens when those abnormal cells don't just grow in that same area, but now they're going through the bloodstream and attaching to other organs throughout your body. And that's when cancer becomes deadly. Normal cells have what's called density-dependent inhibition. And this is a really important term. What this means is when they all start touching each other, they just form a single layer. When they reach this certain density, they know to stop growing. So this shows remove two of these cells in a petri dish and they're going to grow to meet each other, but they're not going to keep growing. They also have anchorage dependence. So normal cells actually have to be attached to some type of basement membrane. So you see these not only are growing in a single layer, but they're attached to something. They can't just break off and go travel somewhere else and grow. They need to be attached to some type of membrane. So that's called anchorage dependence. So cells must be anchored to a substratum of some sort. But cancer cells are both density independent and anchorage independent. So they don't have to be anchored to anything, and they'll just keep growing and forming this mound. Cancer cells have escaped the cell cycle controls. Okay, so they don't exhibit density-dependent inhibition where they stop growing when they touch each other. They can go somewhere else in the body and divide. And also, it's important to realize that it doesn't matter what the cell type is. So if it started off as an abnormal cell in your liver, if that becomes metastatic, it means that that cancer cell can now go grow on another organ that's not a liver, even though it's not the same cell type originally. Also, cancer cells are immortal. They can just keep on going through cell division forever. Normal cells cannot do that. Normal cells have a certain number of times they can divide and then they stop. They're resistant to apoptosis. They induce blood vessels to grow to them, as I mentioned before, and they can spread to other sites. I know I'm kind of rushing through these slides, but if you want to read them in more detail, definitely look at the PowerPoint presentation that I'm going to post. There are two types of cancer genes. And really the difference is, I'm going to try to draw this on the board and I hope you can see it. So tumor suppressor genes, we talked about those in the cell cycle a little bit. The big one that you might recognize the name of is your p53 protein. That p53 protein is coded for by a gene, and that's one of your tumor suppressor proteins. It's involved in that G1 checkpoint. It signals the immune system to destroy a bad cell. In other words, tumor suppressor genes put on the brakes. Okay, so they halt the cell cycle when something is wrong. If you have a defect in this, then you're not putting on the brakes. You're not stopping the cell cycle when a cell's DNA is bad. Oncogenes are the opposite of tumor suppressor genes, and that is oncogenes. This is like putting on the gas. This is the accelerator. Oncogenes accelerate cell division under normal conditions. If something goes wrong with an oncogene and it's accelerating cell division when it's not necessary, then the cells start dividing out of control. And that's what cancer cells do. So this is putting on the accelerator and not stopping. We know of at least 400 somatic cancer genes, but obviously there are probably more than that. So tumor suppressor genes, again, normally prevent bad cells from dividing. And when something goes wrong with them, then bad cells are allowed to divide. Whereas oncogenes normally promote or accelerate cell division. And when something goes wrong with them, it's just dividing out of control. These are just some quick cancer stats. It's pretty horrible. The most common cancers are breast, lung and bronchus, so that's bronchial tubes, prostate, colon, melanoma, bladder cancer. You can just read that list there. Men, 43% of cancers in men are prostate and lung and colorectal. And for women, it's breast cancer, lung cancer, and colorectal. So you can just read those stats. I won't test you on these stats. I'll never ask you specific numbers like this because of course they're changing every day, right? But I just think it's important to kind of have an awareness of this. The cancer mortality rate is slightly higher among men than women. And ethnicity affects cancer rates in the United States as well. Cancer survivors are increasing. The way we treat cancer today is so different from how we used to for most cancers. Hopefully in one of your higher level classes, they will talk to you more about cancer therapies, but a lot of the new cancer therapies and hopefully the future of cancer therapy is to not poison people with chemotherapy anymore, but to do things to your own immune system to help your immune system detect and kill cancer cells, especially modifying your T cells, programming them to recognize the cancer cells and go kill those specific cells, because chemotherapy is a systemic poison and it kills a lot of cells. Most chemotherapy drugs target cells at metaphase of the cell cycle. Metaphase is really the longest stage of the cell cycle, even though it seemed short when we gave that explanation. Metaphase, I know it's hard with the glare on my screen right now, I'm sorry. Metaphase is when that M checkpoint happens. And the premise is cancer cells are constantly dividing. So at any given time, a lot of them are going to be at metaphase. Remember, a normal healthy cell is mostly at interphase. But any cell that's rapidly dividing, like your skin cells, your hair cells, fingernails, cells of your stomach lining, those get affected by chemotherapy because a lot of them are in metaphase as well. The idea is freeze that mitotic spindle or do something during metaphase to keep those cancer cells from dividing. But if we can have therapies that really focus on having your immune system attack and kill the cancer cells, then that doesn't affect the healthy tissue. It's only affecting the cancer cells. And that's kind of the future of a lot of cancer treatment. And of course, that is very specific to the person and the genetics of their tumor. So there's a huge focus on DNA analysis of each individual person's tumors and engineering T cells that can destroy that person's specific type of cancer, the specific cells that that person has that are abnormal, because everyone's cancer cells are different. So even just within one category of cancer, say lung cancer, the genome of everyone's cancer cells is a little bit different. So really tailoring that treatment to that specific person is very, very important in the future of cancer treatment. If you have any questions about cancer treatment or cancer cells, we're not spending a lot of time on that in this class, so please don't hesitate to ask me. But otherwise, thank you. This was a long lecture. You stuck with it, and that's great. And now remember, this would have been 3 videos. Now you're done in one, and that's all.